Hi everyone, my name is Kendrick. For the past couple of years I've been working in the Rothman lab here at UCSD in the biology department. And most recently, under the mentorship of Dr. Pradeep Joshi, I've been working on a project trying to characterize the role of Ice1 in regulating germline apoptosis, proliferation, and differentiation. So, apoptosis, also known as programmed cell death, is a universally conserved and genetically regulated form of cell death. It is one of the most critical processes and thus one of the most highly regulated. There are both pro-apoptotic proteins and anti-apoptotic proteins, and the balance between the two are critical in regulating proper development and homeostasis. However, when this balance becomes misregulated, you can have some really severe disease pathologies. For example, if you have too much cell death, you can end up with mass atrophy or cell death of the brain tissue, such as Alzheimer's disease. And on the other end of the spectrum, if you have too little cell death, cancerous cells can divide uncontrollably and eventually form tumors that inhibit normal functions. So there are a plethora of diseases associated with aberrant apoptotic signaling: AIDS, neurodegenerative disorders, ischemic damage, autoimmune diseases, cancers, and the list goes on. So by identifying and characterizing the proteins that regulate apoptosis, we can better understand the exact molecular pathogenesis of these diseases. In addition, with this information, we can actually develop novel therapies that actually target these misregulated cell regulators. So specifically, I'm studying the cell death pathway in C. elegans, which is really well understood because it's actually discovered in this model organism. On the left here is actually the image of the organism that we work with. It's a small soil-dwelling nematode worm, and as a fully grown adult, it's about 1 millimeter in length. But despite its small stature, its contribution to biomedical research has been immense. So the canonical cell death pathway in C. elegans are these series of proteins. And basically these proteins act as cellular actuators that turn on and off certain events, eventually causing cell death. And importantly, all these proteins in the C. elegans pathway have equivalent proteins in humans, also known as homologs, which is important in trying to relay the relevance of our research back to human disease. In fact, there was a study done in which they eliminated all of the C. elegans SED9 cell death repressor and actually replaced it with a human gene, and they saw a rescue of that repressor function. So this really drives home the point that our research may be really relevant to human diseases, although we're only working with a worm. So the specific apoptotic regulator that I'm working with is called inhibitor of cell death 1, or Ice1 for short. And initially the question was, what is Ice1's role in regulating apoptosis? However, upon further investigation, we actually found that Ice1 is also regulating proliferation and differentiation. So the new question is, what is Ice1's role in regulating all three of these very different and distinct processes, and how does that relate back to the human homolog called beta-NAC? So in order to better understand the exact cellular function of Ice1, what we did was played around with the expression levels. So we knew that proper Ice1 expression was necessary for normal development, so we wanted to see what happens if we either knocked out 100% of the Ice1 protein, if we decreased Ice1 through a knockdown, and what happened if we overexpressed Ice1. And today I'm specifically going to be talking about the results from the Ice1 knockdown. So this was done through a technique called RNA Interference, or RNA for short. And basically how this worked is it decreases protein expression by preventing the translation of RNA into protein. So what we did is we took our gene of interest, Ice1, and transformed it into a special type of E. coli that actually produces double-stranded RNA. We then feed this E. coli to the worms and looked at the progeny, looked at the worms. So when the double-stranded RNA actually enters the worm, it gets uptaken by the cellular machinery. And the cellular machinery then takes one of these strands and uses it as a template for site-specific targeting of the Ice1 gene. So the cellular machinery then finds the Ice1 mRNA and it actually degrades it, so you get less protein expression overall. So specifically, I'm looking at the effects of Ice1 on cell death, proliferation, and differentiation within the C. elegans hermaphrodite gonad, which is highlighted here in the blue, and there are two distinct arms of the gonad. On the bottom is a more detailed image of this left gonad arm, And to orient you, in this region right here is really important. This is the distal end of the gonad, and basically this is where all your mitosis or proliferation is occurring. So all your cells are generated within this region, and as you move along the gonad, you can actually see the cells begin to differentiate nicely into oocytes and sperm in this proximal region, and this is where fertilization occurs and the embryo formation. So what we found was that when we decreased Ice1, we actually saw an increase in cell death. So for this assay, we used a dye called Cyto12, which basically preferentially stains dying cells brighter than living cells. So as you can see here, in our normal worm, we saw about 4 to 5 corpses on average. However, in our Ice1 RNAi worms, we saw this number jumped up about 4 to 5-fold to about 20 corpses. And in addition, the localization of the corpses shifted from this more distal region to a more proximal region. So we also wanted to look at the effects on proliferation, and what we found was that when we decreased Ice1, we also decreased cellular proliferation. So in this assay, we used a dye called DAPI, which basically stains the nuclei so we could count the total number of cells. So as you can see here, our normal worm and worm with decreased Ice1 actually began with about the same number of germ cells in an early developmental stage. However, as you can see, the decreased Ice1 worm had a slower proliferation profile, and you ended up with only about half the total number of germ cells as normal. Surprisingly, even though our Ice1 RNAi worms had a decrease in the total number of germ cells, there was actually an increase in proximal tumor formation. So in our normal worm, you would expect to see nicely oocytes and fertilized embryos within this proximal region. However, in our decreased Ice1 worm, what you see is just a mass of what appears to be undifferentiated cells. So to more closely characterize this, we used antibody markers for a better analysis. So we had an antibody marker against, we used an antibody against a proliferation marker called phosphohistone H3, and we used another antibody against differentiated sperm protein to see if any differentiation was actually occurring. And again, we used the blue DAPI dye to stain the nuclei to see it. So to orient you, this is the distal region of a gonad, and this whole panel of images is our normal worm. So this is the distal area, and as you move along, it actually continues in this bottom panel down to the proximal region. And as I mentioned earlier, proliferation only occurs within this distal region, and you should never see any proliferation markers anywhere else within the gonad. And in addition, you see the sperm protein nicely localizing to where fertilization occurs, which is all normal. However, in our Ice1 worm, although you see proliferation markers in this distal region, you also see aberrant proliferation markers throughout the entire gonad, which could be an explanation as to why we're actually seeing this tumor formation. In addition, we also saw differentiated sperm protein, which was a major question for us because a lot of these worms were actually sterile, meaning that they didn't produce viable offspring. So we wanted to see if there was any sperm production at all. So clearly, Ice1 is regulating many different aspects of development. It has both apoptotic and non-apoptotic functions, and when we decreased Ice1, we saw an increase in cell death, a decrease in cellular proliferation, and issues with differentiation. So currently right now, we're also analyzing the genetic knockout strain and our overexpression strain, and once we figure out what's going on, we can begin to elucidate the mechanism by which Ice1 is functioning. So, although these results are promising, it's also a cautionary tale. So, for example, many anti-cancer drugs function by triggering apoptosis. But from this research, we showed that sometimes if you trigger apoptosis, you can also increase cell proliferation in the spatially different region, which would be the opposite of the desired outcome. So, hopefully, with this knowledge, we can be wary and better understand the exact role of cell death regulators to develop more effective therapies against a wide range of diseases associated with aberrant cell death signaling. So now I'd like to thank Dr. Rothman and Dr. Joshi for mentoring me, in addition to all the other Rothman lab members, Erica, Maria, and Kevin for all their support and help this summer, the Arnold and Mabel Beckman Foundation for their generous funding through the Beckman Scholars Program, and I'd like to thank you now for listening, and I'd be happy to take any questions.